everyone, I'm Akshitha Suresh and I'm pursuing my MSc in Biotechnology from Krista Jayanti College, Bangalore. This video was prepared as an activity that was assigned to us by our teacher Dr. Manikandan, Faculty Department of Life Sciences, Krista Jayanti College, Bangalore. In this video, I'll be talking to you all about the Maxim and Gilbert method of DNA sequencing, which is also known as the chemical method for DNA sequencing. I'll be giving you a very brief introduction about DNA sequencing and then talking to you all in detail about the Maxim and Gilbert method. I'll cover the procedure, advantages, disadvantages, and the applications of this method. So to start with, what is DNA sequencing? DNA sequencing is nothing but the method or a technique which is used to determine the sequence of nucleotides that are present in the DNA. Any information that is required for the growth or the development of an individual or an organism, it is encoded within the DNA of the particular organism's genome. And therefore, DNA sequencing is fundamental to genome analysis and understanding the biological processes in general. Now, DNA sequencing is of two types, the chemical method and the enzymatic method. Chemical method is the Maxim and Gilbert sequencing method, and the enzymatic method is the Sanger sequencing method. This chemical method is divided into 4 steps. So the first step is denaturation, which is followed by radioactive labeling, chemical degradation, and then gel electrophoresis. So let's talk in detail about Maxim and Gilbert method. This method was developed by Alan Maxim and Walter Gilbert in 1976. This method is also known as chemical degradation method for DNA sequencing. In this method, we can sequence the DNA fragment by using specific chemicals. Now, the nucleobase specific partial chemical modification of the DNA and subsequent cleavage of the DNA backbone at the sites that are adjacent to the modified nucleotides help us in sequencing the DNA properly. So to start with, the first step is denaturation. In this step, we denature the double-stranded DNA to single-stranded by increasing the temperature or the pH. And then we run the single-stranded DNA fragments on the gel. Like we all know, due to the lesser molecular weight of the lighter fragments, they move further than the heavier fragments. And these heavier fragments are often bands that have large number of purines, which is adenine and guanine. The next step is radioactive labeling. In radioactive labeling, we radioactively label one 5' end of the DNA fragment that has to be sequenced by a kinase reaction using the gamma phosphorus. Then we cleave the DNA strand at specific positions using chemical reactions. We can use either of these chemicals, which is followed by piperidine. So dimethyl sulfate, it selectively attacks on the purines, which is the adenine and guanine, and hydrazine will selectively attack on pyrimides, that is cytosine and thymine. All the radioactively labeled fragments are then put into 4 reaction test tubes. So each reaction test tube will be used for each base to identify each base. Now, for adenine and guanine, that like I said, for purines in general, we use dimethyl sulfate with high temperature and NaOH. So in this tube, this reaction mixture is capable of denaturing adenine, which means it'll either denature adenine or adenine will be present in a very less quantity. So this tube is usually used for the detection of guanine. The next one is just dimethyl sulfate with HCl for adenine. Then next composition is for the pyrimidines, which is hydrazine and piperidine. So here it will be like a very less amount of thymine present or no amount of thymine present, and a very less amount of cytosine present or no amount of cytosine present. And in the next one, we use it for cytosine, so that will be hydrazine with NaCl and piperidine. So these are the reaction mixtures that are added to each of these tubes respectively for the bases guanine, adenine, thymine and cytosine in the order. Now after the chemical degradation, we get these nucleotide sequences that are specific to these chemicals with the bases. Uh, and after that, we subject them for gel electrophoresis, where the fragments are electrophoresed on an acrylamide gel and they are separated by their sizes. All these fragments from each of these four tubes are poured into the gel, and the four wells that are made on the gel, in the first well it'll be like the fragments from the first tube, second well from the second tube, and so on. These fragments, like I mentioned before, they separate on the gel according to the size, and due to their lesser molecular weight, the smaller fragments move further than the larger ones. And after placing the radioactively labeled film on top of the gel, radioactive labeled fragments would emit a spot on their position. This is after the gel electrophoresis. Now, what are the advantages of this technique? No premature termination due to DNA sequencing takes place. Then, homopolymeric DNA runs are sequenced as efficiently as the heterogeneous DNA sequences. It can be used to analyze nucleic acid structures and epigenetic modifications of DNA. The stretches of DNA can be sequenced which cannot be done by enzymatic method. Then it can be used to analyze DNA protein interactions like DNA footprinting. The next I will be talking about is disadvantages. So it is not a widely used method. We widely use is the Sanger sequencing method. Then it is difficult to make the Maxim Gilbert method sequencing method kits, kits based that can work based on this principle. Then it requires extensive use of hazardous chemicals, like I mentioned before. The read length decreases from incomplete cleavage reactions. We are using radioactive and toxic chemicals here, and it is difficult to scale up this process and cannot be used to analyze more than 500 base pairs. What are the applications of DNA sequencing? Applications of DNA sequencing include conditions where it provides an information regarding evolutionary relationship. It helps in gene identification and mapping. It helps in the identification of mutations that have taken place, and it also provides the sequence information. These are the applications of DNA sequencing, and these are the references that I've used to prepare this PowerPoint. Thank you.